A warm welcome again to the Kiel Imaging Seminar. It is great to have you all here today, and we have a fantastic speaker. It's my pleasure to introduce our KIS seminar speaker to you. It's Professor Franz Schilling. He studied physics at the University of Würzburg and then at the University of California in Berkeley. He obtained his PhD in Munich at the TU and focused on nuclear magnetic resonance. During his postdoctoral research at the University of Cambridge, he deepened his interest in molecular imaging, which he still pursues today. And since 2017, he now leads an independent research group at the Klinikum rechts der Isar at TU München. In the last year, he was appointed associate professor for biomedical magnetic resonance at the TU München. And before we start, to the audience, please wait with your questions until the end of the presentation. And with this, Professor Schilling, the stage is yours.
Yeah, thank you so much, Jos, for the kind introduction, and good evening, everybody, and really a pleasure to be speaking here in the KIS imaging seminar. It's quite an impressive number of speakers and list of speakers who have spoken before me, and I think it's really great to interact with all of you today. And it's my pleasure to give you an introduction to what we're currently up to in Munich, what research we are doing, entitled currently today, Imaging Life and Death. And I'll tell you about novel MRI methods for detection of cellular function and metabolism. Just as a brief intro, where are we? So our lab is basically located in the middle of Munich. You can convince yourself why our TUM University Hospital is called rechts der Isar, because if you look at it from this angle, it's actually directly right of the river Isar. Here is the Bavarian Parliament, and here is the clinic with our imaging labs. See our group here who contributed to all the results you're going to see today. We've already seen a brief CV about where I've been working on. So my background is in physics. I studied in Würzburg and Berkeley, then did a PhD in chemistry at TUM with a short postdoc in England, and then came back to TUM as a research group leader. So we now have quite a multidisciplinary team here in Munich, consisting of physicists, chemists, medical doctors, and we have quite an interesting working environment to come upon. So our infrastructure here in Munich is split in two parts. We have a nuclear medicine department where we do basic research with instruments. You see preclinical instruments, polarizers, also preclinical PET and clinical PET instruments, anti-translatum, where we also support studies in PET, MR, and SPECT for larger animal studies and more screening purposes. We also interact with the physics and chemistry department in Garching, have access to high field NMRs and X-ray CT, high resolution CT. So coming to the title of today's talk, Imaging Life and Death, and I want to quote one of my favorite biochemists here, Jacques Monod, who really commented on the difference between systems that are living and dying. And the main difference that he mentions here is that basically the essential unit that you need to create life is a cell. And as soon as a cell is broken, as soon as the structure is broken, the system cannot live anymore. Doesn't need to saying that, of course, metabolic features can only happen in the inside of a cell. And we will also get back to this artwork that you see here, which resembles water molecules in the membrane here, later on when we talk about MRI methods that can detect membrane ruptures. So what are we doing in my lab? So we have basically three research areas that we focus on currently: metabolic imaging, pH imaging, and diffusion and exchange. And I will get into all of these during the talk. What is basically consistent is that we try to develop novel imaging biomarkers for cellular function based on MR physics, which means sequence design, molecular MRI sensor molecules to basically approach new biomarkers and implement those in preclinical and clinical studies. We apply a multimodal imaging approach where we also work with other techniques like PET and SPECT to investigate those biological and medical research questions. So let's start with metabolic imaging. And some of you, and most of you, I guess, are already familiar with the principle of hyperpolarization. which is a method that we use in MRI to boost our sensitivity. The drawback at thermal equilibrium is that effectively only 5 in a million proton spins contribute to the MRI signal. But this corresponds to the situation where you're basically just looking at the tip of an iceberg. The good news is that you can artificially create a hyperpolarized state, which then renders those spins invisible that don't contribute to the effective net magnetization. And there are now a couple of techniques that can be used, that we use, that also many people in the community use. The one that is most heavily applied is dissolution DNP, where you use extremely cool temperatures and then you transfer polarization from electron spins to the nuclei. You heat the entire sample up, do a dissolution, and then inject it readily into animals or also in patients, as has been demonstrated. More recently, also other techniques employing a different source of polarization, in this case, parahydrogen and its spin order that can be transferred as magnetization via hydrogenation reaction to a molecule, and then also polarize nuclei such as carbon-13 spins in respective molecules and lead to polarization that after separation of, in this case, a sidearm that has been used to polarize the sample has been cleaved off. In addition to this, reversible exchange via the Saber technique and the catalyst that is being applied leads to a reversible hydrogenation of a molecule and can also lead to polarization of the molecules so that we can apply them in biomedical applications. This increase in sensitivity was really demonstrated by dissolution DNP by the landmark paper by Jan-Hendrik Ardekje-Larsen in 2003, where you see one very impressive experiment from this paper shown here. A sample consisting of 60 millimolar concentrated hyperpolarized urea acquired in a measurement time of 1 second compared to the same sample non-hyperpolarized, which took a measurement time of 65 hours. And you see the difference in signal-to-noise ratio immediately, which really directly shows you the impressive enhancement in sensitivity that you can get with those techniques. So in our lab in Munich, we use this technique, dissolution DNP. You see a polarizer here, which is a hyposense instrument that can polarize the sample. We can then extract them via this tubing line that either leads to a tabletop spectrometer for quality control or directly leads to a preclinical scanner where we collect it and then can inject it into animals. More recently, we've also worked with a parahydrogen-based polarizer provided by Envision, working with the parahydrogen technique, also for hyperpolarizing biologically relevant molecules. We are preparing clinical trials with the parahydrogen-based technique for 2027 and are looking forward after the approval of the regulatory authorities, which hopefully comes soon, to also inject patients with techniques in the near future. So what is now good news for the community at this stage is that with all of these techniques, dissolution DNP, FIP, and Saber, you get very decent metabolite images. Here, just for comparison, this is healthy mice that were scanned with the 3 different techniques with exactly the same sequence, slightly different sequence parameters. But you can appreciate that the images that in this case were acquired with the 3D BSSFP sequence here give fairly similar quality of the images, showing the bolus of pyruvate entering in here in the mouse and then the conversion, the metabolic conversion to lactate in the kidneys, which is a real-time metabolic observation of metabolic products here in vivo, which would otherwise, without these polarization enhancement techniques, not be possible. We've also compared these recent developments using parahydrogen-based techniques to the established dissolution DNP ones. And here you see an example where we have a rat that's bearing a mammary adenocarcinoma that you see here in the proton image with a large tumor here in the field of view. And now, after we've injected pyruvate, and I will run the video here, we can follow the 2 substrates, the substrate and the product, pyruvate and lactate, directly within the tumor, collecting a metabolite time curve. If we inject the same animal a second time, in this case with parahydrogen-based polarized pyruvate, and compare the 2 time curves next to each other, you almost don't see any difference. So in this case here, confirming for a single animal that the two techniques can be applied readily, one after the other, with no difference in terms of metabolite saturation or any other toxic side effects. And in a larger study confirming this also, we've seen that both with the DNP polarized pyruvate and the Fib polarized pyruvate, we obtained similar metabolic quantitative readouts, such as the area under the curve ratio measured by Fib or DNP, which give a very good correlation. We've also altered the order of injections. So first and second injection was randomized, either Fib or DNP, but didn't observe any difference. So this was all good news in applying this technique further in animals. So one sequence that we currently use, that we've developed in our lab, is a special variation of a balanced steady-state free precession sequence. The BSSFP sequence is quite efficient for acquiring hyperpolarized signal because it really reduces RF consumption and makes the acquisition fast and efficient. What we do is we use an additional trick here, spectral selectivity. So we shift our radio frequency pulse with regards to its offset to do off-resonant excitations, basically switching between one metabolite and another metabolite. This really permits short TR times because we can work with short and broad off-resonant RF pulses, which can give us a high spatial temporal resolution. So the images that you have seen previously, they were acquired with this special variation of the BSSFP sequence that you see here. Additional factors that contribute to the signal-to-noise ratio are, of course, your hardware. And I want to show some unpublished results here that we have conducted with cryogenically cooled coils. So that means we have a coil from Bruker that is cooled with gaseous helium to roughly 30 Kelvin. Also, the preamplifier is cooled, which for a receive-transmit coil really can enhance the SNR quite dramatically. So here you see comparisons of a cryoprobe CSI image compared to a surface coil with the same diameter and a standardly used 31 millimeter inner diameter volume coil. These are the spectra that you acquire, and you can directly appreciate the increase in SNR here by using this cryogenically cooled coil. If you compare these two coils, the thermal coil and the cryo coil, which have a very similar profile in terms of the receive and transmit B1 fields, you can appreciate that you get, depending on the distance from the coil, SNR enhancements in terms of a factor of up to 8-fold. So quite a lot of gain in SNR that we can use and exploit, even though, of course, we have limitations because so far we can only work with transmit-receive surface coils with a very inhomogeneous B1 profile. Nevertheless, we have exploited this, and here you see images from a mouse brain, in this case static CSI, demonstrating that we can detect pyruvate, lactate, alanine, also bicarbonate in the brain, in this case with a Fib polarized pyruvate sample that was polarized up to 20 to 30% every 10 minutes, and that we could then use further for metabolic studies in the mouse brain. With this 3D spectrally selective BSSFP sequence, we get a 1 millimeter isotropic resolution where you see the individual metabolites, pyruvate and lactate here resolved in 3 spatial dimensions, 1 temporal dimension, and here all across the brain. This now allows you to also characterize individual regions of the brain, which are outlined here, and their respective metabolic time curves that you see here. And we see, for example, big metabolic differences here between the olfactory bulb and the thalamus, factor of 2 difference in the AUC ratios, meaning that this is a technique that can really characterize and quantify metabolic differences within the healthy brain and also, of course, within pathologies. Another area that we are interested in is in vitro systems, and this is basically a platform that is looking at patient-derived organoids that are implanted on a fertilized chicken egg. The idea here is to look at human-derived cancer cells and look at their tumor growth in an artificial environment. And here you see basically in this fertilized chicken egg where the chick embryo is already developing and the tumor is implanted in the system, that it's being vascularized and that it's also growing in a region here, so that we have a perfect or a very good resemblement of a human tumor within an artificial system that we can then study and that we can also study metabolically. So we spent quite some time also now on optimizing the setup for metabolic imaging, and this is work done by Miriam that she presented the last ISMRM, where we've shown that with a dedicated, in this case, inductively coupled booster coil that was developed by our cooperation partners in Ulm, we can very sensitively detect pyruvate and lactate signal within this tumor region, within the fertilized chicken egg, and characterize its metabolic behavior within the system. So we hope that in the future we can also use this system to study patient-derived tumor samples with regard to different treatments and characterize their metabolic response. Of course, besides pyruvate, there's a lot of other probes around in the hyperpolarized community, and of course, this is always a subjective selection, but I've focused on a couple of those probes here, mainly besides metabolism, where you can study glycolysis and energy metabolism, pyruvate to lactate conversion, and also different metabolic products and educts in the TCA cycle. Also reactive oxygen species, for example, via DHA, gluconolactone, or pH extracellularly and intracellularly via bicarbonate, ZOMPD, zymonic acid. Those are pH agents we have developed in the lab that I will go into, and that we also currently quite heavily work on because we're quite interested in measuring extracellular pH. And with this, I would come to the second topic of the talk today, which is pH imaging. So first of all, why do we want to image pH? Why is this important? Here we can state that it's really relevant in many different pathologies, quite globally: inflammation, ischemia, renal failure, and cancer. And it is a parameter that's currently not being possible to image routinely in the clinic, even though it's quite important in those all different pathologies. One example is the acidification of extracellular pH in tumors, which is basically happening because from the intracellular space, the tumor cell exports lactate together with a hydrogen ion and acidifies the extracellular environment, which helps the tumor also to progress, to invade, and also makes it more resistant to therapy. So it's really one of the features that a tumor needs to supply its further growth, and therefore can be a potential target in order to attack the tumor. So the approach that we have taken is we have synthesized two pyruvic acid derivatives as pH sensors, which luckily have an acid dissociation constant that lies in the physiological pH range, which makes those molecules by NMR detection so extremely sensitive for pH. And this is zymonic acid here on the left, which we've introduced first, in this case already in a deuterated version, which helps us in terms of maintaining the hyperpolarized signal because it increases the carbon-13 T1 time by quite a bit. And here ZOMPD, this open linear structure, also labeled with two carbon 13s at position 1 and 5 of the carbon. Those two compounds, they can be readily synthesized from carbon-13 labeled pyruvate and its esters with a high purity, and they're also non-cytotoxic. How do they work? So this is a small animation that shows how the NMR resonance frequencies of zymonic acid change with pH if you alter the pH. So in this case here, going to higher pH, and the chemical shift basically because of this acid dissociation constant in the physiological pH range changes because of the deprotonation and protonation that affects the NMR resonance frequencies at these two carbons. And then this pH sensor and its frequency shifts with respect to urea, which is a non-pH dependent reference compound. It's very similar for the OMPD compound. However, with the difference that the OMPD1 peak shifts much less. So it's well better suited as an internal reference in comparison to zymonic acid. And in most cases, you don't need an additional independent reference such as urea. So you can basically just work with an internal reference and inject just one agent instead of two agents. So if you titrate the pH and look at the NMR chemical shifts, you see that for OMPD, this changes with respect to pH, and you can deduct the calibration curve from this and then back calculate the pH. What's very favorable for this compound is that the T1 times, they're very long. So at 1 Tesla and in D2O, they exceed 2 minutes. In blood, at 1 Tesla above 1 minute, and then at 7 Tesla drops down to around 30 seconds without degassing and filtrating the radical of the compound. So those very long T1 times and also the good polarization level that we can achieve so far with the solution DNP make those compounds quite favorable with regards to their imaging properties. Here are some examples what you can do with this. So this is pH mapping in the kidney, which can clearly detect an average pH here in the rat kidney. But on the other hand, we also know that there are 3 renal compartments in the kidney, the cortex, medulla, and ureter, that have different physiological pH values responsible for the filtration properties of the kidney. And this is something that we can also resolve. First, I want to show this here with a technique which is quite interesting in terms of the acquisition strategy, because we can do single voxel spectroscopy with this technique. So we've done pH imaging with a semi-laser sequence. So we've actually really used 90 and 180 degree pulses to excite our spectra with high spectral resolution in a scheme that basically takes care of not saturating the individual voxels from the preceding acquisition. So you need basically to create a 3D acquisition matrix that makes sure that from the previous excitations and refocusings, you're not saturating your magnetization. This allows you basically in a 3D volume to excite several voxels with high spectral resolution and also with very good spatial localization of these individual spectra. And then from the spectra, you can calculate individual pH compartments, as for example here in the kidney of an animal. What we've also done is that we have combined zyanic acid and OMPD with imaging readouts. Here we see an acquisition that has basically acquired the perfusion of OMPD via acquisition with a BSSP 3D sequence on the C1 carbon of the compound in the kidney, showing the perfusion in the kidney. And then after that, fit CSI measurement, taking the spectral resolution to calculate an average pH map from both the C1 and the C5 carbon, combining basically a perfusion, filtration, and pH measurement that just takes 60 seconds within one measurement. So here you see a comparison of this method in healthy kidneys versus a rat that's having hydronephrosis, so a pathological method, in this case induced by a cancer. And we can already see in the perfusion videos, and here indicated by the white arrows, that there's basically a blockage of the filtration process in the kidney. So the agent, in this case OMPD, doesn't perfuse toward the center structure of the kidney, which we can then also calculate and see in the glomerular filtration rate. We can quantify from the individual compartments in the kidneys. So it really indicates this poor transport into the renal pelvis indicated by this tracer and a strongly impaired glomerular filtration function. This was not visible in the pure blood flow measurements here from the perfusion of the agent of the cortex. In addition to that, we get the pH measurement. As I said, we can calculate the mean. We can nicely already see the deviations from physiological pH of 7.4 here in the 2 kidneys. But since we have a spectral measurement, we can actually decompose this further. So in the cortex and also in the surrounding tissue, we see a compartment at pH 7.4 present in both the healthy and the hydronephrotic kidneys. And here you see its spatial extent. Then going to the next spectral compartment, which is separated in frequency here, we can see that the medulla compartment at pH 7 also present in both of the states, the healthy and the hydronephrotic animals. But if you then look at the renal pelvis, you see this very strong acidification of the renal pelvis in the hydronephrotic state, which in terms of the pH value, you only see if you do the spectral decomposition, which is, in my opinion, one of the advantages of those frequency-based pH sensors, because in your voxel, you can detect different pH compartments relying to or underlying basically different physiological structures, as here in this case the cellular function of the kidney in different compartments. So we see this distorted acid-base balance in the renal pelvis, and one question we had is this something that's relevant? And for example, if you compare this to standard conventional proton MRI, where you see also a kidney dilation in the hydronephrotic state compared to the healthy state, and also a small difference with diffusion-weighted imaging, we see that the effect size of the pH measurement and the glomerular filtration rate is much bigger compared to the others, which means that this is probably a complementary additional readout that is really useful in characterizing disease states, which gives us additional information that we can extract from our images. Now, the question that we also asked is this a technique that we can apply clinically? And for that purpose, we have set up a study on a clinical system, a 3 Tesla PET MRI, with animals, which in this case were cats that were included in the study based on different inclusion criteria. Basically, we are looking for malignant mesenchymal tumors, a curable disease, and a good overall conditions, so that the owners got a free care, surgery, and follow-up of the measurements. And what we then did in the PET MR is we tried to get a metabolic characterization of the tumor environment using dynamic PET measurements, injecting fluorine 18 labeled F-meso, a hypoxia tracer, carbon-13 labeled xymonic acid for measurements of pH, carbon-13 labeled pyruvate for measurement of pyruvate lactate conversion as a measure of metabolism, and then also DWI in addition to measure the cellularity of the tumor. So from this study, we have recruited 18 cats on the clinical scanner. We got very decent readouts of pH maps, acidification, metabolism, and hypoxia. And on the bottom here, you see how the different subtypes of the tumors, which were characterized by pathology, histologically differed in terms of their acidification, so the acidification relative to muscle, the area under the curve ratio in terms of the pyruvate lactate conversion, and the Ki, which is the kinetic rate constant for the accumulation of F-meso correlating to hypoxia. So it was very interesting to see that we can differentiate those subtypes, and also that if you look at, for example, this group of fibrosarcoma here at the beginning from grade 1 to 3, that they didn't really differ too much in terms of their hypoxic state, but in terms of acidification and area under the curve ratio, there was really a change and an increase with higher grade. So in this case, we can already see from this that potentially hypoxia is not the driver for acidification in those tumors. It could be rather excretion of lactate that is driven by a different metabolic feature of those tumors. And it seems really that in this case, for the fibrosarcoma, we see a correlation. Of course, we have to be careful because it's small numbers for those animals between the area under the curve ratio and the acidification in this case. This was quite remarkable for those fibrosarcoma of different grades, but not so much for the others. One feature that we also saw that by analyzing the overall survival of those cats is that the acidification from all of these parameters was giving us the best prediction of overall survival of those cats, which also means that it's probably an interesting parameter to look at in terms of characterizing the progression of tumors in the future. And also good news is that the spectral resolution at 3 Tesla that we get from these pH sensors is enough to characterize the acidification and the pH changes, even also under more challenging conditions and a smaller absolute chemical shift at lower fields. So with that, I'd like to come to the last topic of my talk, which is about imaging transmembrane water permeability. And right now, there are basically two applications that we have explored for this. One of them is to look at changes in water permeability because different expression of water channels, such as aquaporins. And also another one, which I'm going into more detail here in this talk, is that we can use it for detection of early cell death. So early cell death in comparison to late stage cell death is different in a way that the cells are not disintegrated, but that you have still defined cell walls and small structural pores or holes in it that are very difficult to detect, and that, for example, with a conventional diffusion-weighted imaging sequence and ADC measurement are very hard to detect, because you basically would observe no change in cellularity at such an early stage of the cell death process. What are we using to determine those permeability changes? We're using a filter exchange imaging sequence, which is based on the principle of a double diffusion weighted sequence that was really pioneered by Paul Callahan in the early 2000s in a spectroscopic measurement, and then later by a group in Lund pioneered to be applied in an imaging setting with reduced acquisition parameters. The basic idea behind this is that you measure an apparent diffusion coefficient with and without a diffusion filter that alters the magnetization of your spins in the extra and intracellular state. In the intracellular compartment, the water molecules, they are more confined. That means they have a lower diffusivity. That means if you apply a diffusion measurement or diffusion filter in the beginning, they will still be magnetically active, whereas the ones that are in the extracellular compartment, fast diffusing, will be saturated or attenuated. So in this little scheme that you see here, we see the water molecules in the outside being saturated and the ones in the inside being activated and then diffusing outside. So depending on in which timing or after which mixing time you measure your apparent diffusion coefficient on this filter, you'll detect a different diffusivity. And this we can measure. We can measure the ADC in dependence of the mixing time, and will observe a relaxation of the ADC if, due to permeability, these two compartments equilibrate. The readout that we measure here is the so-called apparent exchange rate, which is directly proportional to the permeability and which gives us a non-invasive readout via magnetized water of how water permeable and how the water is exchanging across a cellular membrane. And with that, we can get back to the picture from the very beginning, where we see here magnetically activated water molecules exchanging with the extracellular space, and of course also going the other way in the direction of the saturated, attenuated water molecules going the other way around. And because of this exchange process, we can measure pores or holes that you see in the cellular membrane, which are a very early indicator of cell death. We've also done a lot of validation measurements of this method. Here in this case, it's really important to look at repeatability of AXR and reproducibility in yeast, which is a very simple two-compartment system. Here you can see that we can measure AXR in the yeast, also image it across different phantoms. You can increase the water exchange rate by increasing the temperature, which you can nicely see resembled here at different temperatures. You see the different relaxation curves at different temperatures, going back faster at higher temperatures, and correlating directly linearly with the AXR. We can also inhibit the aquaporins in the yeast, and we see directly a reduction in the AXR. So all of this confirming that this is a method that we can nicely apply in simple two-compartment systems to measure the water exchange rate. So we've taken this further, and here you see acute myeloid leukemia cells after necrosis. We've destroyed the cell here by isopropanol and then confirmed this measurement by FACS, so fluorescent activated cell sorting. What is quite remarkable here is that the XR measurements by NMR, they detect this increase in permeability at very low concentrations, 5%, 10% of isopropanol already, which is a sensitivity threshold that also the FACS method has. So here we see the first change at 10% of isopropanol, where we see early cell and late cell death with two stainings, in this case, the fluorescent markers, annexin 5, and also propylium iodide. Whereas we don't see a strong change in terms of the apparent diffusion coefficient, really here telling us in this in vitro setting that this might be a promising marker to look at early cell death. Confirmed was also that at 10% isopropanol, here with electron microscopy, the cell really gets those initial pores. There is this membrane damage already occurring at these concentrations. So this must be the reason why we see this increased permeability in the cells. We've then looked at different processes of cell death, in this case here necrosis, apoptosis, necroptosis. and have correlated the AXR with the percentage of annexin 5 and propydium iodine stained cells, so the total number of dead cells. And we saw a very nice correlation of AXR with the fluorescent markers for all of these cell death processes. They were induced in a completely different way, whereas for the ADC, no change here with regards to the number of the stained cells. Again, confirming that we're looking at these early damages compared to the late stage damages that we can observe with DWI and ADC measurements. Then the next step was to look at this in an animal, and here we've done a study where we've combined filter exchange imaging with DWI and also a hyperpolarized marker, in this case, fumarate to malate conversion, which is a marker of early cell death in a way that the mitochondria is damaged, and then the enzyme fumarate is released, resulting in the production of lactate. And again, here we saw a correlation between the AXR, the ADC measurement, and also the AXR and the fumarate to malate ratio. Then the question is, can we use this now to detect early treatment response? And here in a study that we've conducted together with Karen Brindle at the Cancer Research UK site, we've treated EL4 lymphoma with etoposide. Again, with ADC, no change at 24 and 6 hours after the treatment, whereas with AXR already after 24 hours, there was a difference observable in the images leading to enhanced water permeability at this stage. This was confirmed by another study in a colorectal carcinoma that was treated with a TRAIL2 agonist. Again, here after 4 hours, already changes in permeability that we think would come from an early treatment response in this model, leading to porous membranes that were damaged because of the therapy. Then with this technique, one question is, of course, can we translate it to humans? And because this is a technique that doesn't involve any exogenous agent, it basically just simply uses water molecules as the contrast mechanism and standardly available DWI sequences. We have looked at a test case where we've recruited patients with uterine fibroids that were embolized. And we did an MRI measurement, including filter exchange imaging before embolization and then 24 hours after. And here you can see the AXR maps in these uterine fibroids before and after, already indicating a change in the AXR, a change in permeability, really showcasing here that this is a technique that could be directly applied in humans. Now, in these two patients that I show here, we see the distribution of AXR before and after embolization really shifting to higher AXR values, indicating the cellular damage, which we see in both of the patients. So you see the first patient here, increase in AXR across the fibroid. Here as well, increase in the fibroid. At the same time here, the ADC is decreasing also in both patients. So what's going on here? What we have here after embolization is a process very similar to stroke, where you have cell swelling. So you have an increase in the intracellular volume, intracellular space, leading to a decrease of the ADC in those regions. So in this case, really two processes that are going on in parallel here. On one hand, cellular damage, increase in permeability. At the second time, also a decrease in diffusivity, meaning there's also cell swelling going on. So with that, I'd like to summarize my talk. So we've seen techniques for metabolic imaging, methods for 3D spectral, spatial, temporal imaging, and also several options for polarizing carbon-13 nuclei like FIB-SABR and DNP. We've seen novel hyperpolarized pH sensor molecules that we developed and applied simonic acid and ZOMPD with the specialty that they can really resolve subvoxel pH heterogeneity because of their frequency-based pH readout. And last but not least, we've seen how with diffusion and exchange measurements, we can assess the function of aqueous pores and also membrane permeabilization methods that can be used to detect early cell death. So the further goal of this is to translate those methods to the clinic and really make use of those novel techniques. And with that, I'd like to thank the entire team behind this, who's really responsible for acquiring data. You've done a great job, and thank you so much for your contribution. I'd like to thank all my collaborators who contributed to this, and I'd also like to make you briefly aware of a conference that we're organizing in Munich, 23rd of March, the Translational Hyperpolarized Imaging Network and Conference, with quite a lot of exquisite speakers discussing translational aspects of hyperpolarized metabolic imaging. So if you have time, come by. There's also shuttle buses that are being organized to EMIM if you want to join the EMIM conference in Ljubljana, but you can find all about this on the webpage. So with that, yeah, really thanks a lot for your attention. Thank you for your contributions here, and I'm open for any questions now.
Yeah, thank you very much for your nice presentation. It was a blast. Please go ahead if you have any questions. You can also write them in the chat and I will read them out, or you unmute yourself and ask the question as you like.
A very nice talk. I had two questions. One of them is, do you feel that with these techniques, there is still a role for phosphorus 31 NMR spectroscopy in the study of metabolism, in particular that it is, of course, endogenous and 100% naturally abundant. It's been used for many years in pH measurements, although distinguishing between intra and extracellular pH is pretty challenging.
Yeah, so simple answer, definitely yes. So I mean, there's many options you can use phosphorus spectroscopy for. I'm thinking of muscle exercise measurements, ATP measurements, which is really an exquisite technique. With regards to clinical applications, besides like the basic science, biological questions, it might be a bit more tricky because of the sensitivity issue and the long scanning times. But I still think there's plenty of room in terms of research and preclinical studies for phosphorus 31.
Okay. And my other question has to do with apoptosis. Do you feel that it's possible to distinguish between necrotic and apoptotic cell death and also necroptosis using a diffusion sequence?
Yeah, that's a very, very good question that we were also thinking about. I want to briefly pop up that slide that I had about comparing those different methods of cell death that we see here. And it was quite surprising for us, actually, that all of these forms of, let's say, rather more controlled cell death in a way of talking about apoptosis and necroptosis versus uncontrolled cell death in terms of necrosis are at an early stage very similar. So at the stage where those first porous holes or fractures in the membrane occur, they're actually quite hard to distinguish from each other. I think the option to distinguish them would be to look at them over time, to look at the dynamic process and to look at how they differ at different temporal stages, which is something that we have so far not done, but which would be interesting to look at. So I think in terms of characterizing the changes dynamically, both via XR, DWI measurements, and also measurements of intra versus extracellular spaces, that could be an option to distinguish them.
Yeah, I know that many years ago there was some attention to detecting apoptotic cell death through inversion of the cell membrane and differences in mobility that occur in some of the lipid molecules. But I don't know that that's been followed up thoroughly.
Interesting point. Yeah, that would be also something that could be combined with those techniques. I think the more biomarkers we obtain also in a dynamic fashion, I think the easier it would be to separate and characterize those different processes.
Thank you very much.
I also have a brief question to the slide basically that you're showing here. I was just curious, do you have any insights, like, for example, if you think about hyperpolarization in this instance, hyperpolarization often demands fast... my microphone is up. Maybe you understand me better now. A question for this slide.
Yeah.
So I was just curious if you have any insights on hyperpolarization, how it correlates with what you observe here, because hyperpolarization often demands fast transfer from the extracellular to the intracellular space, right? And you could imagine that now the higher porosity of the cell from necrosis, apoptosis, whatever, leads to faster uptake of the hyperpolarized tracer, for example, and thus maybe to higher metabolism, or that that the cell death on its own decreases metabolism. I was just curious if you had seen any correlation data here between hyperpolarization and AXR in some example.
So we have not done the experiments here with fumarate and pyruvate at these early stages of cell death. We have looked at FDG uptake, and it's also a figure in the supplementary part of the publication, where we basically see that the probe that we use here, water, is exquisite or extremely sensitive to those early porous changes. So, for example, at these isopropanol levels of 5 to 10% that we see in the earlier slides, we already have the penetration of the water molecules at a very, very small fraction of damage to the cell, which you cannot see, for example, in the glucose uptake, and I suspect also not in the fumarate or pyruvate uptake at this stage. So the water molecule, also because of its very small size and because of its very high sampling and abundance, is very exquisite and very sensitive in sampling this. But yeah, we have not done exactly the same study with pyruvate and fumarate, so that could be an interesting point.
Okay. And concerning the measurements that you did at 3 Tesla of ZOMPD, I was just curious if, like you showed that T1 was multiple fold longer at the lower field strengths, right?
Yeah.
Comparing 1T versus 7T. And for these kinds of measurements that you're performing, T1, I think, provides a great benefit. So I was just wondering if, like, the sensitivity limitation of the lower field strength was basically outweighed by the longer T1 in the case of ZOMPD, if you know what I mean.
Yeah, so there's two things to look at. I mean, you definitely get longer T1 times, which is a benefit. On the other hand, of course, you lose some kind of like spectral resolution in terms of characterizing your pH compartments. So it definitely kind of like goes in opposite directions here. In this case, we definitely benefited from the longer T1 at 3 Tesla, which, but also we have other issues. For example, in the cats, the sensitivity of the coils, the availability of pulse sequences on the clinical scanners, which also have to be taken into account. So a direct sensitivity comparison is not possible. We are also exploiting, of course, other possibilities to extend the T1 times. For example, we're currently working on the deuteration of the OMPD compound, similar, for example, to xymonic acid, where the deuteration increases the T1 even further. So there's probably still some room in terms of enhancing the sensitivity at the clinical field strength.
Okay, thank you. Are there any other questions? Then I maybe have one more follow-up question. Which of the techniques that you showcased here are you most excited about translating into the clinic? It seems that AXR maybe is the closest, as it doesn't involve like all the legal requirements. But talking about excitement for the technique.
Well, they're all exciting. I would say they're all exciting. I mean, with the AXR, it's great that you can directly move to patients and that you just use water, which really also you can directly see this from the reaction of the patient, right? If you don't inject the substance, it's very easy to convince a patient to do such a study. It would be probably similar with deuterated compounds that you just take by oral gavage, that you just drink. But on the other hand, such a pH measurement, I think that the hyperpolarized technique is really ideal for that because you get a very quick measurement, you get a pH readout within 1 minute. And I find it quite fascinating that within 1 minute you can extract all this information, such as perfusion, filtration, and pH, and then you're done with the measurement. And this could also be clinically valuable. So yeah, I am excited about all of those techniques. If one of them makes a difference in the clinic, I'm happy.
Yeah, totally agree. Thank you so much. Yeah, maybe last opportunity to ask a question about the nice presentation. You can also write them in the chat. Yeah, okay. Then I thank you very much for this great presentation. It was very insightful.
Yeah, thanks so much for the invitation. It was a pleasure.
Very nice having you. I hope everybody has a nice evening, and hopefully see you soon at some point, maybe at a Think conference in March.
Great. Yeah. Have a nice evening, everybody.